Welcome to Ask and Learn.
Hello students, here we are being asked various multiple choice questions, and the first one is bind to and inhibit ribosomes. The correct option is option C, which is tetracycline. Another question is bind to and inhibit enzymes that make folic acid. The correct option is A, which says that trimethoprim and sulfa drugs. Another question is two E. coli isolates could belong to two different strains. Is it true or false? It is true. Option A is correct, which says that true. Another question also is which of the following is selectively toxic? The correct option is option A, which is all of these drugs, which include trimethoprim and sulfur drugs, penicillins, tetracyclines, and fluoroquinolones. They are all selective drugs. Another question also is the gene ARG-C, ARG-B, and ARG-H code for enzymes, which are proteins, of the arginine synthesis pathway. As we know that the arginine is an amino acid, which is a monomer. Now select the most probably wrong statement about these three genes. The correct option here is option D, which says that when arginine molecules are plentiful in a cell, these genes will be transcribed. Another question also is what prevents DNA from getting tangled as it is replicated? The correct option is option D, which says that DNA gyrase. Another question also is anti-chikungunya virus antibody molecule. Regarding this, the correct option is option E, which says that are made by the B lymphocytes of a mouse that's been infected with chikungunya virus. Another question also is what promoters are made of RNA? The correct option is option B, which says that this statement is false. Another question also is we can reduce the number of drug-resistant bacterial strains by using antibiotics as little as possible. The correct option is option A, which says that this statement is true. Another question also is what is the biggest difference between different types of serological tests such as slide agglutination assay, fluorescent antibody assays, and ELISA? The correct option is option E, which says that the method used to determine if antibodies are binding to antigens. Another question also is, substitution mutation can make a gene longer or shorter. The correct option is option B, which is false. Another question also is, a bacterial chromosome has one origin. That's true or false? It is a true statement. Another question also is, broad spectrum drugs are selectively toxic. This statement is false. Another question also is, which of these is the smallest? The correct option is option E, which says that nucleotide among the given options is the smallest. Another question also is, during DNA replication, what breaks hydrogen bonds between base pairs in a parental DNA strand? The correct option is option E, which says that helicase. Another question also is, a mutation where dash nucleotides were inserted into a protein encoding gene would be expected to cause the least significant change in the protein the gene encodes. The correct option is option C, which says that 3. When 3 nucleotides are inserted into a protein coding gene, then there is a possibility of having a least significant change in the protein. Another question also is, bind to and inhibit enzymes that make peptidoglycan. The correct option is option C, which says that penicillins, which are also known as beta-lactam antibiotics. Another question also is, by using penicillin, humans created penicillin-resistant staphylococcus. The correct option is A, which is, this is a true statement. Another question also is, a dash has the most genes. The correct option is A, eukaryote. Another question is, bind to and inhibit DNA gyrase. The correct option is E, which is fluoroquinolone, which also includes ciprofloxacin. I hope you found the video useful. Thank you.